Hi, I'm Crystal McLaughlin. I am an assistant professor here in chemistry, and I study microbial proteins. I'm a biochemist slash biophysicist, and today I'm going to talk to you a little bit about my research interests and what we do here in my lab at Vassar. So I'm really interested in microbes at the molecular level. So microbes including bacteria and viruses, and sort of what is happening, what are the processes at that level. So one of the reasons that I got interested in microbes was this complex relationship that humans and microbes have. What do I mean by that? Well, in the last couple of decades, we've really started studying something called the human microbiome, which is all of the microbes that live in and on us. So they include bacteria, they include those viruses, they include fungi, archaea. Really, when you walk around, you are in fact mostly microbe from a cellular point of view. For every one human cell that is on you right now, you have about 5 microbial cells. So it's a 5-to-1 ratio. And these microbes aren't just hanging out and catching a ride with us. We've actually co-evolved with them for hundreds of years, and in fact they play a very big role in human health, keeping us healthy. Now, when we think of microbes, when you think of a virus or bacteria, you probably think of getting sick, of some sort of infection. And so those microbes are a subset of our microbiome and other things that we'll come in contact with, and they're the pathogenic microbes. So they're the pathogens, the ones that cause a disease. However, we need to sort of have this balance between protecting our helpful symbiotic bacteria, which do really cool things like break down food for us and extract nutrients. Just as an example, in your gut you have about 1,200 different species of bacteria living there, and they help us get nutrients and food all the time. And when they go away, like say you take a round of antibiotics, which kills many of them, you feel sick, and it's because you've lost that helpful bacteria trying to maybe kill a pathogenic bacteria. So the question then becomes, how do we protect the helpful bacteria while still being able to treat the pathogenic bacteria? And that is sort of the overarching question that I try to answer in my lab by figuring out these molecular details of the microbes, and in this information trying to learn, can we find some way to really target the bad bacteria, the unhelpful ones, and keep our good ones safe. So when we think about treating these pathogenic bacteria, these microbial infections that we have, we think about drugs. So how do you treat how do you treat pneumonia? You take a drug. How do you treat Lyme disease? You take an antibiotic. And so the discovery and development of antimicrobials, which is the general class for all of these drugs that treat any microbial infection, really was a turning point in human health. So the biggest example of that I can think of is the discovery of penicillin. When penicillin was discovered by accident, as you probably heard that story, it really became this great discovery because it allowed us to not die from very simple bacterial infections. Penicillin could cure simple bacterial infections. We were no longer dying from that. And it was the first antibiotic in wide use, and since then we've discovered and created many more antibiotics. However, another thing you may have heard of is that with our increased use of antibiotics, we do have increased antibiotic-resistant strains of bacteria. So those two go hand in hand. So how do we treat those? How do we create and get better antibiotics is another problem facing the field. Now, when we also think about a different kind of drug, which I put here as an example, ritonavir, which is an antiviral. So ritonavir is used as part of a drug cocktail to treat HIV and AIDS. Ritonavir was in fact developed in a lab, so very different from the discovery of penicillin, which was an accidental, fortunate discovery from a fungus, whereas we were able to have some knowledge about HIV, the virus that causes AIDS, and we used that knowledge to craft and create a drug that could help treat it. And so this second way is another very helpful way for creating a drug. While we still look out in nature to find drugs like penicillin and other antibiotics and antifungals, etc., we are also in the lab trying to figure out what's happening at the molecular level in the microbes and then creating drugs to target them. Now, I just want to remind us a little bit about some biology, which you may have heard of, which is often called the central dogma of molecular biology, which is just a general idea for the flow of information in the cell. And why am I talking about this? Because it has to do with what do these drugs target. When a drug is working, a drug like penicillin, a drug like ritonavir, what's happening? So if we look at the central dogma of molecular biology, we have DNA, which I'm showing here in blue, and it's transcribed in a process into a messenger molecule called messenger RNA, which is shown in purple. And so the messenger RNA is then used and translated to make a protein. And so proteins are really the workhorses of the cell. They do many, many cellular processes. For example, enzymes carry out pretty much all of the chemical reactions in the cell. There are some proteins that are structural that create the cell wall and different compartments in the cell. Proteins have a wide range of jobs, and so they are in fact usually the target of a drug. Ritonavir was created to target HIV protease. That's the name of the protein, and it's because we had knowledge of that protein and we used that knowledge, that molecular knowledge, to make the drug. Now, penicillin kind of was the opposite way. We discovered penicillin worked, and then later on we figured out how it worked, and it in fact, spoiler, binds to a protein, and that is how it does its job. So looking at these proteins is what I do in the lab, and trying to figure out any molecular details that could help us figure out better treatments is the major goal of my research and research interests. Now, this whole process of creating a drug by using the molecular details is actually something called rational drug design. So it's defined as using knowledge of a target, in this case the protein, to design a drug. You can, of course, design drugs to target other things, but proteins are usually the main target. Now, I want to give you an example of rational drug design that's happening right now. We are in the middle of a pandemic, and in fact, COVID-19, which is going around, is caused by a virus called SARS-CoV-2. And scientists all over the world are trying to find out as much as we can about SARS-CoV-2 so that we can create treatments for it. Now, some people are trying to design a vaccine, and some of us are looking for drugs as well, too. So those are different things. So rational drug design is being employed now. So how do we get the molecular information that we need about, say, the virus in order to try to design a drug? Well, first, you can start off by taking a picture. These are very common pictures that you see. You can see the entire virus particle here. This is an electron micrograph, so you can see the virus particles are colored in red, yellow, and then they have sort of a green outer layer. Now, this is not enough detail for us to design a drug to target something. We need protein level detail. So if we zoom in a little bit, this is now a cartoon representation of the cross-section through the virus, and you can start to see more details. For example, at the center of the virus, you see this purple sort of squiggly thing. That represents the DNA. And then on the outside, you see the purple and pink sort of magenta. Those are called spike proteins. And every virus has spike proteins that are on the coat. And those are a very common place that we try to design drugs for. But we still can't design a drug yet. That is not enough detail. So if we zoomed in just a little more and looked at just one spike protein so that we could see the structure of it, here, this is what scientists are using right now to try and design drugs. So this is an actual structural image of the SARS-CoV-2 spike protein that was solved a few weeks ago. It's shown here in many colors because it has many different parts. But all of this information, this is atomic level details. We can see where all the atoms are positioned. We can see the shape of the protein. We can try to find unique features that allow us to create targeted drugs to this virus to stop it from going forward. And in my lab, this is what we do. We create these images of the proteins, these atomic level detailed images that hopefully will help other scientists develop treatments for drugs. In my lab here at Vassar, we use X-ray crystallography as our main tool for capturing the structural detail. It's a very cool, actually I've said cool too many times, but I think it's super cool. It's a really neat biophysical method that involves, as the name implies, X-rays as well as crystals in order to probe matter and see what the molecular structure is. Now, how do we use X-ray crystallography to provide atomic level structural detail of protein? The first step is actually getting enough protein or DNA, whatever biological molecule you want to investigate, or whatever kind of matter. We need a lot of it. And then, in the case of proteins in our lab, we have to coax the proteins into forming a crystal. So the crystals I'm showing here are sort of diamond shaped and they look a little colored. They're very, very tiny. They're on the micron level. But these crystals aren't very different from crystals you might interact with in everyday life, like, say, salt crystals or sugar crystals. It's just that instead of it being made up of salt and sugar, we have very, very tiny crystalline material, and it's made of proteins. And of course, a lot of water, because all biological molecules need water to survive. Now, we then take those very tiny crystals and we expose them to a beam of X-rays. The beam of X-rays will interact with the crystalline matter in that it gives us something that looks like this, which is called a diffraction pattern. This is our X-ray data, so these sort of black dots. And here at Vassar, we actually have our own X-ray source, which is very exciting to use whenever we have students. And seeing sort of students gather X-ray data for the first time is always a really fun experience. So it's really great to have it right here at Vassar so we can try to figure out the structure of our proteins and other matter right here. Now, we can take this diffraction pattern and we can use that to create something called an electron density map. This map tells us the position of every single atom in the material. And it's shown here as this sort of blue wire frame. Now, into this frame, we can build a model of all of the atoms and build the entire protein. And then what you see in like textbooks or maybe even the picture that I showed before of the SARS-CoV-2 spike protein is the model, the final model of the protein, which just represents different features in the protein, sort of like this sort of curly thing, which is an alpha helix, and arrows, which are beta sheets. And then this model enables scientists to pick out unique features, to understand how the protein works, and to find new ways, maybe, for example, designing something that fit right into a nook like of this protein at the bottom. And that's how you would use this molecular level model to create a drug. Now, as I mentioned before, in my lab, we're really interested in microbial proteins, and we in fact study microbes from a couple different systems. And the question we're really trying to answer is, can we target selectively a bacterial strain? How can we do that? So can we learn enough about a microbe to just target the one microbe? For example, if you had, say, pneumonia, can we just take an antibiotic that only targets pneumonia, leaving the rest of the microbiome intact, not killing your helpful bacteria? Currently, that's not an option, as many antibiotics are very broad range. So learning more molecular details about these microbes, our hope is that it helps us to create these treatments in the future. So I'm going to tell you a little bit about one project in the lab that we're working on that has to do with antibiotic resistance. You may have heard of Staphylococcus aureus. It's also known as staph. Many people get staph infections. And normally, you can treat a staph infection by taking an antibiotic and then it's gone. Staph actually is part of the normal healthy microbiome of about a third of all people. It doesn't actually cause any infections. Some of you probably have it in your nose and on your skin right now. And so normally it's totally fine. So this picture that I'm showing up here is a picture of cultured staph, and you can see it sort of looks like what I think of like grapes. They actually color it purple a lot. And so it's this globular bacteria, and like I said, it doesn't usually do very much in the normal microbiome. It's just hanging out, being chilled. However, it is an opportunistic pathogen. What this means is, let's say you got a bruise, an abrasion, or some sort of cut. If staph got in there, it could cause an infection. So it really only causes infection when there's an opportunity, which is why people often get staph infections in the hospital when they're already injured or are sick. Now, a bigger problem is MRSA, which you may have heard of, MRSA, and that stands for methicillin-resistant Staph aureus. And MRSA is a big problem in hospitals. It causes about 80,000 infections per year in the US, more in the world, and is considered a serious level threat pathogen by the CDC. And this is because our normal round of antibiotics cannot treat MRSA. It is resistant to many antibiotics. And when it causes a staph infection, it makes that staph infection now likely fatal. So figuring out more about Staph aureus and how staph works at the molecular level, particularly around antibiotic resistance, would be really helpful for creating treatments or maybe even just stopping these antibiotic resistant staph infections. Now, antibiotic resistance in many different bacteria are actually harbored in the cell and taken care of by these specialized proteins. So it's a special piece of DNA, and you need these specialized proteins to keep the antibiotic resistant genes in the cell. And that is where this project comes in. We're looking at one protein that helps keep resistance inside of Staph aureus. The idea is, if we learn enough about these proteins, maybe we can stop them from doing their job, and then staph can become non-resistant once again and able to be treated with regular antibiotics. So the protein I'm going to talk about is called COP. Its full name is Staph aureus PSK41 COP, which is a mouthful. We're just going to refer to it as COP for now. So what we've done in the lab so far on this project is we've crystallized it and we have solved its structure. So first, I'll show you a picture of the crystals, which are very cool. So we grew these here at Vassar, and as you can see, there's a little scale bar on the crystals. They're sort of square, a little bit diamondy, and they are the yellow things in the drop. And they are on the order of microns. As I mentioned, these are very small crystals, much smaller than, say, a salt crystal, which you can see with your naked eye. We need a microscope to see this. These crystals are about 600 microns they grow to about 600 microns in length. Now, we take those crystals, we exposed it to X-rays, and then we were able to solve the structure of COP, which I'm showing here in the blue. And so this is the first molecular picture of this protein COP from Staph aureus that has ever been taken. No one knew what it looked like before. We only had ideas. Now, I can show you a little more. Let me show you a video of it in 3 dimensions. So just this video is going to play, and it's going to turn around, and it's going to show you all of the features that we can see. It's not just a forward snapshot when we get this structure. So as you can see, it has sort of these wings, and we think that the wings are how it binds DNA because we think that is its job, to keep those genes inside of the bacteria. Now, one of the other things, like I said, we think these wings are where it binds to this outer area of the protein. We think this is where it binds DNA because we think that is its job. We can also use some more details that we get from the structure to try to figure this out. So we can represent the structure in a different way. So now what I have shown is a different representation of the structure. It's like a surface representation, on top of which I've superimposed the image we were looking at before, so it's in the same orientation. Now, if we take a look at this surface representation is colored in white, red, and blue. The blue is where there are positive charges, and then the red is where there are negative charges. So we can map that onto this picture that we have. Now, when we turn the protein around sort of 90 degrees, we can see that it has this very long track underneath of unbroken positively charged area. DNA is very negatively charged, and so this makes sense. Many DNA binding proteins have these long positively charged tracks where they can bind and hold on to DNA. So we think that's where it binds. However, we don't know, so of course more is needed. More research is needed to really, truly figure that out. So that is the next part of this project. And right now in the lab, we have a student that has grown some new crystals of it. And as you can see from the picture of these crystals, they are much, much smaller than the ones that I showed before. What this means is we can't actually get data from them yet. So this is an ongoing project. We have to figure out now how to get those crystals big enough to put in front of the X-rays to get our data. So a new student will be working on this coming up this fall. So in summary, microbes can be helpful or harmful, and developing new drugs can be aided by structural knowledge, and this process is called rational drug design. X-ray crystallography, which is the main technique we use here in my lab at Vassar, and currently the most popular technique for getting these pictures, gives you this atomic level structural detail of biological molecules, allowing you to perform rational drug design and really just learning a lot more about the molecular processes of biological systems. So thank you so much for listening. I always like to thank all the students that do all of the work in my lab, and they make working here such a pleasure. So those are pictures of them, and thanks to everyone to help with that. And then on my next slide, I have some further resources. So if you found this very interesting and you even just want to go back to basics, learn more about what is a protein, or even more about crystallography, I've put some helpful videos and resources here for you to follow up on. And of course, if you have any questions, I am very happy to help answer them via email. Thank you so much.